just like you want to back up your data, you also want to back up your physical preps of samples and things. And so one of the most important things to back up is your plasmids. And so these are the circular pieces of DNA that say have the instructions for making a protein. And you often go through a lot of work doing molecular cloning, doing subcloning, doing site-direct immunogenesis in order to get a plasmid that has the sequence that you want and it doesn't have any extra mutations and things. And to ensure that these plasmids are correct, you typically send them for sequencing. So I like whole plasmid sequencing. They'll send you back the sequence of the plasmid, you check it against your reference, what you're aiming for, and see if you got it good. Now, sometimes you don't. And so typically what happens is you send multiple of these clones, multiple of these plasmids for sequencing. And some of them will come back good and some of them will come back bad. When you get those results, you want to separate the good tubes from the bad tubes. Stick the bad tubes in a to be deleted box. And so you can kind of think of it as the trash can on your computer where you have to actually go and physically delete it from your trash later, but in case you kind of accidentally threw it away or something. And so in case there was a labeling mix up or something, you don't want to throw those tubes away right away, but you kind of want to separate them, maybe make some red markings over them so you don't mix it up, and you separate it from the good ones. And now what do you do with those good ones? You obviously want to keep the good ones, but you don't just want to keep them, you want to keep versions of them for backup. And you want to keep the backup in a stable form that will allow you to make lots more of it in the future. So at this point, you have this purified plasmid, and you want to make it so that you can make more of it. In order to make more of it, you've got to stick it into bacteria to make more of it. And you stick it into these cloning cells of bacteria, something like DH5 alpha, to make more of it. Now, you might be thinking, well, didn't I just purify it out of DH5 alpha? So I have that whole plate of these colonies that contain it. Well, you have a plate of colonies that contain kind of the plasmids, but you don't know if those plasmids are the good ones. And so remember, some of those were probably the bad ones. And so you need to stick this plasmid into new DH5 alpha or other cloning cells in order to get kind of a whole population of these good clones, where each of the colonies on the plate should be one of the good ones. And then what you want to do is you don't want to just store them in colonies on a plate, you want to store them for longer term storage. So what you do is you take one of those colonies, you pick a colony, grow up an overnight culture, just like you would have been doing a mini prep. And in fact, I often do a mini prep of this so I can get more of the purified plasmid. But what I also do is I make a glycerol stock of it. So take 500 microliters of the overnight growth and I mix it with 500 microliters of 50% glycerol. I mix it up and I freeze it. I typically do 2 of these glycerol stocks. Now in the future, if I want it, I can basically just take it from the glycerol stock, streak out a new plate, and voila, I've got more colonies containing my plasmid of interest. I can then mini prep them out, and maybe I want to check the sequence again just in case, but it should be good to go. And once you know that you have ones that are good to go, then you can go ahead and toss those bad tubes that you'd saved in your kind of temporary trash. You're also going to want to mark the plate that you had, the original cloning plate, and then the plate with the good ones that you did when you did the new transformation, because you don't want to mix those up. So you're going to want to very well label the plate that was the version from the cloning and stick that kind of in a different stack, kind of like your temporary trash as well, because typically you're not going to use that plate again because a lot of those colonies might be bad. If your goal is to express protein from this plasmid, you're going to want to transform it into expression cells, something like BL21, DE3. But you're still also going to want to transform it into those cloning cells like your DH5 alpha, and that is how you're going to actually want to store it for future plasmid getting. So for the long-term storage of these plasmids, the best way to store them is in those cloning cells like the DH5 alpha in the form of glycerol stocks, so not just colonies on a plate, but actually those glycerol stocks in your -80 freezer. You don't want to just store them long term in the BL21. You can store them in the BL21 for making protein, but typically I just retransform them to get kind of better expression typically. But for the DNA fidelity, for keeping that DNA sequence really what you want, which is kind of the whole goal of what we've been talking about, then you're going to want to store it in those cloning cells. And then if you need more of the plasmid later, you can go plate it out and purify more of the plasmid from the colonies that you get from that transformation of that plasmid that you know is good that you got in the beginning. plasmid from a colleague or a friend or even from some repositories, it's still a really good idea to sequence it. Not that you don't trust your friend, but it's really easy for kind of mistakes to pop up in the plasmid over the course of the plasmid's lifetime and things. And so even if it's a plasmid that you've been working with for a while, you might want to periodically go and check the sequence, especially if you're getting weird results with your plasmid that you're using. But the first time you use a plasmid, before you actually go and do some experiment with it, you want to make sure that the sequence is what you think it is. Sometimes you don't even know the sequence of the entire plasmid if you order it from a place like Addgene. They often just send you the sequence of the region of interest, which is like the insert, the gene for making a protein you want, say the coding sequence for that protein. And although that might be the region that you care most about and you're most worried about there being mutations in there, that doesn't mean that mutations elsewhere in the plasmid won't impact things. And there could be mutations elsewhere in the plasmid, but you wouldn't know about it if all they're giving you is the sequence of the region of interest. Additionally, if you're wanting to do some more cloning, like subcloning and mutagenesis and things on the plasmid that has the insert, well, then you're going to want to know the sequence on either side of your insert and around the insert and things. And so you want to know the whole sequence of the plasmid. And so then it's a really good idea to go and sequence the entire plasmid so you have that information. And you can go ahead and send a bunch of samples for sequencing at the same time. If you've got a sequencer, you can hook things up to do multiplexing and do your own sequencing. That'd be really cool, but we just send our samples for sequencing. And because we want the whole plasmid sequenced, but we're able to do it in one run, we don't have to kind of waste a bunch of money doing different Sanger sequencings to cover in overlapping regions the whole plasmid. We just send the whole plasmid, get one read, and then we have that information. Now, sometimes that information might be correct, but it doesn't match what you have as the correct sequence in your kind of like reference. And so sometimes when you get a plasmid, you might think it has something, but it actually has something else. And actually this happened to me once where I got this plasmid and it was like this whole different protein than what I was actually intending to get. And so I'm really glad that I checked the sequence in that case. But sometimes it's more minor things and there are kind of slight differences in the spellings that don't actually impact the protein that gets made or things like this. And so sometimes those little changes are okay, but sometimes they're not, and it's better safe than sorry. Sequence your clones, keep the best, toss the worst, but keep them in those temporary trash before you really toss them. So separate the bad from the good. Keep the bad in the to be deleted, to be trashed, but don't throw it away until you're sure it is what you think it is and that is not what you want it to be. And then you take the good stuff, you retransform it into DH5 alpha, you grow up some more, you make some more plasmid. If you want more plasmid, you purify out more plasmid. You make glycerol stocks, so I typically do 2 glycerol stocks per plasmid. Now you don't have to, if you find yourself in that situation where you go and you use the last of your plasmid, don't freak out. You don't have to start over. You've got the plasmid in the cloning cells. You can quickly make lots and lots more of copies of this plasmid, purify them out, and use them. But do be sure that you're checking those sequences to make sure that the versions that you're like backing up are good.